Good evening. Tonight we will be discussing the HIV virus and the acquired immune deficiency syndrome. And tomorrow we will be talking about the rest of the sexually transmitted viruses in general, and all of the infectious agents that are going to infect the genitourinary tract. So the acquired immune deficiency syndrome and human immune deficiency virus is a new virus almost discovered in the United States not before 1981, so it's just the last century virus. And the first scientists who really worked on the HIV virus and AIDS were here a man by the name of Montagnier in France, a French scientist, and an American scientist, his name is Gallo, were the first two scientists to work on the HIV virus and AIDS. And it's very important to know that the advances in tissue culture and growing the virus in cell culture has really contributed to the advancement of the discovery of the virus, its pathogenesis, and lots of research that has been done on this virus, and of course on the effect of antiviral drugs in vitro. So when they have studied here about the HIV virus and AIDS, they were looking in retrospectively into the blood sera that were present in labs all over the world, and they were looking for antibodies to the HIV virus, whether they were present or not. And they figured out that there were no antibodies in the sera, in the blood banks, at least before 1950. So this virus has been discovered after the 1950 in Africa, at least, and in 1970 in the United States. And not before that. There's no proof that this virus has been present in the United States before 1970 or before 1950 in Africa. And we do have two viruses that are associated with AIDS, two serotypes. One, we call it HIV-1, which is the most common one, the first one that has been discovered, and HIV-2, which has been discovered in west part of Africa, discovered in 1985. So this topic and virus has been taken from Shiri's Medical Microbiology. It's chapter 18. You can read it in details and over there. And the HIV virus is part of what we call the retroviruses. That means it has the enzyme of what we call reverse transcriptase that here converts the single-stranded RNA into a hybrid of RNA DNA, and then into a double-stranded DNA. And the double-stranded DNA, then it's going to integrate itself into the chromosome of the cell through what we call the integrase enzyme. And then the integrated virus becomes a permanent finding in the genome of the cell. It goes into transcription and then translation, and produces the different parts of the virus. So it is so important for you then to know the exact structure of the virus and all of the proteins that are going to be involved, as well as the enzymes and their functions. Like, for example, the surface proteins, which we call it the P24, or the glycoprotein 24, or the capsid proteins, because antibodies against these capsid proteins, we use them to make the diagnosis and to follow up those viruses. And it's so important to know the target here receptor of these viruses, which are the T helper cells or the CD4 cells. So these findings are extremely important to know, and we have genes that they are involved in this process of binding, like, for example, the CCR5, we call the chemokine receptor type 5, and the CXCR4. Those are co-receptors that will help in the binding of the HIV virus, the P24 attachment protein, into those specific receptors. So I want you to remember here those proteins, the P24 and the CD4, the CCR5 and the CXCR4 as the co-receptor. And of course the reverse transcriptase enzyme that changes the RNA into DNA, and then the DNA is going to integrate itself into the chromosome. So this is extremely important to know in understanding the replicative cycle. So it's single-stranded positive sense RNA that is going to be converted under the influence of reverse transcriptase enzyme into a hybrid of RNA DNA, then into a double-stranded of DNA that's going to integrate itself through the integrase enzyme into the chromosome of the T helper cell, and then it becomes as a permanent here genomic structure in those. So it's so important to know that if the patient is infected with the HIV virus, it is going to be infected for life, and copies of the integrated virus is going to be produced and infect other cells. So those structures, then the replicative cycle is extremely important to know, and understanding of those makes you understand the pathogenesis and the diagnosis, and what are the structures that we are looking for, and why here we are going to end up with a severe immune deficiency. So the lesson that I want you to know, that the target cells are the T helper cells, بنسميهم احنا CD4 cells, and those they have a fixed number in our body from around 800 to almost 1200 per lambda, and when they are infected, then the number of those cells they are going to be much less, and all the cytokines that are going to be produced are going to be compromised, and then the patient is going to have acquired immune deficiency, and becomes more susceptible to viral, bacterial, and fungal types of what we call an opportunistic types of infections. So patients then they die of AIDS, of what we call of an opportunistic infections caused by infectious agents that normally they do not infect immune competent patient. Usually infects patients with a defect in his or her immune status. That means the patient is usually immune compromised and becomes susceptible to infectious agents that normally they do not infect immune competent people, just infects immune compromised. So it's extremely important then to know these facts, the structural components of the virus, the enzymes that are present, the reverse transcriptase enzyme and the integrase enzyme, protease enzyme, and the function of all of those enzymes, and then the replicative cycle and how the patient becomes severely immune compromised. Because if you understand all of these structural components and key points, then you will understand here the pathogenesis, you will understand how we make the diagnosis, and also you will understand how we are going here to treat those types of viruses. Then this virus, the HIV virus, the human immune deficiency virus, is a retrovirus. That means retro from the word reverse transcriptase. It has an enzyme that converts the RNA into double-stranded DNA. It is also an enveloped virus, so that means it's a delicate virus. So any mild antiseptic agents will be enough to disintegrate this virus or to inactivate this virus. It's single-stranded, positive sense RNA. It has the enzyme reverse transcriptase. Reverse transcriptase, we call it an RNA-dependent DNA polymerase. So it works on RNA and makes DNA. So we call it RNA-dependent. It works on RNA. DNA polymerase that acts and makes DNA out of the RNA. It is a lentivirus. We mean by that it is a slow virus, and one of these lentiviruses, like it's not just the HIV virus, which is 1 and 2, but also we have other oncoviruses like the HTLV1 and HTLV2, the human T lymphotropic virus type 1 and the human T lymphotropic virus type 2, and those are associated with malignancies. So these types of viruses, then they are oncoviruses, which is extremely important to know as well, beside the HTLV3, it used to be called, or the HIV, which is the immune deficiency virus. So this virus, it is so fastidious, that means any mild antiseptic is going to disintegrate this virus, and it requires an intimate contact for infection, which is sexual intercourse, mucous membrane contact in order to enter the body. So it has to do then with sexual activity, homosexual activity, where, as you know, in male homosexuals, the anal orifice is not designed for sexual intercourse, and so mild bruises usually is going to take place, and the virus could have access through the tiered mucous membranes. Extremely important to know that it attacks the T helper cells, or what we call the CD4 cells, and you should know all the cytokines that are produced from the T helper cells, and when those they are compromised or destroyed, then we become immune compromised. It's extremely important to know this fact. So this is the structure of the virus, and you should know it by heart. So it is an enveloped virus, as you can see, and on the envelope you have the attachment protein. We call it the GP, or the glycoprotein here 120, which is the envelope, and the glycoprotein 41, which is the fusion. So this is the GP120, the attachment protein, and this protein on the surface is responsible for the fusion of the virus. So those are present on the surface. And then inside you can see we have a single-stranded positive sense. Positive sense, that means it can act as a messenger RNA when it enters into the T helper cell. So this is the enzyme, the reverse transcriptase, that changes the single-stranded RNA into a double-stranded DNA that has the ability to integrate itself into the chromosome of the cell. And we have also other proteins or markers that sometimes you could see them when we do here some electrophoresis and so on, like the P17, we call the matrix protein, and this is the P24, which is the structural surface protein. We have also here, look at the MHC, which is major histocompatibility complex, class 2. That means here this is present on the T helper cell. And also we have the P7 nucleocapsid as a protein as well. So you have to remember those proteins of the surface, the attachment proteins, and some other accessory molecules, or what we call the accessory receptors. So the receptor is the CD4, and the accessory, the helper, others, which are the CCR5 and the CXCR4. So those are accessory that the virus can bind to. So the GP120 is the attachment protein, as you can see, and the accessory molecules that are going to help. So now we have mentioned then so many different proteins, attachment proteins, as well as a receptor for the virus, and the accessory molecules for the virus that you have to remember those by heart. The accessory, the chemokine receptor 5, and the CXCR4, the chemokine receptor type 4 and 5. They are very important for the binding of the virus into the T helper cells. So it is so important again then to remember those structural proteins and their function. Usually are the ones that are present on the surface that we call them the ENV, and the ones that are present inside like the enzymes as well known the structural proteins. The surface glycoprotein here GP120 is the attachment proteins to the T helper cells. And then we have the antigens that are responsible for a fusion of the virus to the T helper cells, and we call that the transmembrane glycoprotein, the GP41. This is very important to know. And then we have the other membrane associated matrix protein. We call it the P17. And we have also the group antigens. We call it the GAG, capsid core proteins, or the P24. This is where the virus binds to. And then we have the RNA, of course, molecules, and then the protease enzyme that's so important in the replicative cycle. The P9 we call it, or the polymerase RT, and the RNase that's responsible. Find the RNase H that destroys the RNA at the end after it converts into the DNA. So those different then proteins and structures and their functions, as I said, extremely important to know. So single-stranded RNA that is diploid. Diploid, that means it's present in 2 copies, as you can see. Diploid, 2 copies. And then we have the surface proteins like the GP24, and then the enzymes that are the polymerase enzymes, integrase enzymes, the reverse transcriptase enzymes. All they play a major role in the replicative cycle of the HIV virus. So again, this virus then is a small virus. It's about 100 nanometers in diameter. It's diploid. That means we have 2 copies of the positive sense RNA of a single-stranded RNA genome. It has the nucleocapsid and the capsid protein that has the P24, the main protein that's present in this virus. The surface one is the envelope, here proteins. We call it the GP120, which is the main receptor for the attachment of this virus into its the attachment protein into the receptor on the T helper cells. And then we have the fusion protein. We call it the transmembrane protein, glycoprotein 41. This is in the envelope as well. And also we have others like the matrix protein, reverse transcriptase, which is a proteases, and the integrase enzyme. It's responsible for the integration of the double-stranded DNA into the genome of the cell. And beside that, we have other here proteins. We call them the regulatory proteins, that they play a major role in the replicative cycle of the virus. So attachment proteins, receptors, enzymes, and regulatory proteins as well. So the virus then has to attach through its attachment protein into the specific receptors on the T helper cells, as you can see here. And then it has to enter into the cell, and inside, through the effect of the reverse transcriptase enzyme, is going to change from a single-stranded RNA into a hybrid of RNA DNA, as you can see, and then a complete single-stranded DNA, and then a double-stranded DNA, and then a complete double-stranded DNA that it will enters into the nucleus, as you can see, and through the effect of the integrase enzyme, it's going to integrate itself into the chromosome of the T helper cell, and then it becomes as a part of the genome of the T helper cell. And then when it does integrate into the chromosome, as you can see, so here it will go into the transcription process and the messenger RNA, and then the translation process and the different proteins that are needed for the structural component of the virus is going to take place. So copies then of the RNA genome will be produced, and the different structural proteins also will be produced that they will assemble, and then they will go out as a complete virus. So just simply as a summary, it enters then as a single-stranded RNA virus that has the ability to change into double-stranded DNA, and then it does integrate into the chromosome, and through the process of transcription and translation makes the different components of the virus, and then it's going to assemble, exiting outside, taking part of the cytoplasmic membrane, and leaving behind a copy of double-stranded DNA already integrated into the chromosome of the cell. So the name of retroviruses, because of the virus enzyme that changes the RNA into DNA and has the ability to integrate itself into the chromosome and becomes a permanent component of the genome or the chromosome of the cell. So this is a summary of what's going on. The single-stranded positive sense RNA under the influence of the reverse transcriptase enzyme. Then it's going to change into RNA DNA hybrid, and also again under the same enzyme, it's going to be converted into double-stranded DNA. It's so important to know that here at the end of this hybrid, we have what we call the long terminal repeats, that it is going to integrate itself here into the chromosome. So now the double-stranded DNA, as you can see through the long terminal repeats, it's going to integrate itself into the host DNA, as you can see. So the long terminal repeats then here function, the sequence over there, it's going here to integrate itself into the host chromosome, and then it becomes as part of that, and then part of the chromosome, and then the transcription then of the host DNA, here double-stranded, is going to give you a messenger RNA under the influence of the RNA polymerase. And then the RNA polymerase, and then here the messenger RNA is going to go into the translation process, and then different protein parts of the virus is going to be synthesized. So start from then a single-stranded positive sense RNA, ending up with a double-stranded DNA that has the ability to integrate itself into the chromosome. So the process then of transcription and translation is almost similar in many different RNA viruses, that they share the same gene format, where the gag or the structural genes are going to be involved, and then the pull, the polymerase enzyme, and the ENV, the surface proteins that the virus uses for attachment, as you can see. So the gag and the pull and the ENV, here those are the major genes that are here capable of making these structural components here. And whether this is in the mouse HIV, or in the human HIV, human immune deficiency virus, you can see almost the same genes, the gag or the group antigens here, as you can see, and the pull, the polymerase, here enzymes that are involved. And then we have other auxiliary genes that are involved, the VIF, for example, the VPU. Those are the auxiliary genes that they will help in the transcription process. The TAT, the transcription activator genes, here TAX and REX, all of these are the auxiliary genes that they will help in this. And the NEF, for example, the negative, for example, transcription that keeps the process will keep going on. So the VIF, VPU, VPR, VPU, or VPR, for example, or VIF, all of these are the auxiliary genes that they will help in the transcription process. So again, these are the summary of the different genes that are involved in this process. The gag and the pull and the ENV genes, and the proteins that are going to be produced and their function. So in the GAG, for example, they will produce the matrix structural protein and the capsid structural protein, and also the nucleocapsid structural proteins, and then the enzymes that are needed for the processing, like the protease enzymes. So you can see that in the GAG and in the pull here mixture. And then the reverse transcriptase enzyme that converts the RNA into DNA. And then the integrase enzyme that's going to integrate the double-stranded DNA into the chromosome. And finally, the ENV, or the envelope here for the attachment proteins. This is the surface glycoprotein here. And of course, the transmembrane protein that is responsible for the fusion process. So you have to know these genes and the proteins that they are produced and the function of those proteins in the replicative cycle of the virus. So those are the regulatory proteins that the virus uses to regulate this process. The TAT gene, for example, produce the TAT protein, which is the transcription activator here, transcriptional activator process, the TAT. And then the REV here promotes the transport of the unspliced messenger RNA from the nucleus to the cytoplasm. And then we have the NEF from negative here, NEF here, and this down regulation of the cellular here, the CD4 and the major histocompatibility complex proteins. And then we have the VPU and the VPR process. The VPU codes for the protein, VPU protein, facilitates the virus assembly and the release, mainly of the HIV-2 encodes, for example, a protein called the VPX here in VP2 instead of the VPU, for example. And then we have the VPR that codes for the VPR protein, and this one facilitates the nuclear entry in the here non-dividing cells, the arrest, that cause arrest dividing cells and keep it inside as it is. And finally, the VEF here produces the VEF protein, and this one increases the viral infectivity. So the IF from infectivity in certain cell types, it keeps the genome inside. So then, as we see in the replicative cycle of the HIV-1, we see during the replicative cycle, this is the most error-prone process that is going to take place. So we are going to have lots of variations of the HIV virus during the replicative cycle. So we say always that this is the most error-prone and lots of errors during the replicative cycle, during the process of the reverse transcription. So because it is diploid and 2 copies, it goes back and forth through the genome, giving you lots of variations of the genome. So the variability of the glycoprotein 120 antigens, which is responsible here for the attachment. Lots of variations is going to take place. So failure then of the immune system to control the infection, because lots of variations are going to take place. So the immune response that's going to develop against one variable will not protect for the second variable, and so on. So it is the most associated with what we call the error-prone virus. So then during the process of replication, and when the virus integrates itself into the genome, a latency is going to take place. So the function of the regulatory proteins is to maintain this latency phase. So the gene is present in a quiescent manner that is going to keep the virus as it is for a longer period of time without any expression. And then when an activation is going to take place, then it's going to it's going to produce more of antigens that will continue the process. So at the level of the messenger RNA, for example, production and transport, this process is going to take place, or at the level of the messenger RNA translation process. So here the NEF, which is the negative here process, the NEF protein here maintains the latency phase. So it will stay in the body for a longer period of time. And the TAT, the transcription activator, for example, and the REV genes, here those they play a positive role in keeping the antigens in an active format and in transcription here regulator. So in AIDS epidemiology and the the mode of transmission is usually through sexual intercourse, either homosexuals or heterosexuals. And we said that in homosexuality here where it is more common because, as I said, the anal orifice is not designed for sexual intercourse, so bruises and damage could take place that will facilitate the entry of the virus through the damaged anal orifice. Or perinatally, during the labor and delivery process, here where the virus could take place and enter into the circulation, or through blood transfusion, for example, and any body fluid. And from there, because of that, all blood units, they have to be tested for the HIV virus. So clinically then, The primary infection in AIDS usually ranges from asymptomatic to an infectious mononucleosis like illness with up to a few weeks of fever, malaise, arthralgias, and skin rash. A long symptomatic period usually follows, usually years, sometimes 5 to 10 years sometimes, after which the disease AIDS emerge. We call it the full-blown picture of AIDS. The progressive findings directly due to the virus are wasting diarrheas, for example, and a neurological degeneration. The effect of the virus on the immune system, which is the immune deficiency, causes an extensive array of viral, bacterial, fungal, and parasitic opportunistic infections. We said that organisms that usually does not infect normal people, but infect the immune compromised, whose findings are the same or worse than who's seen in patients without AIDS. So this is the classical clinical scenario of an HIV infection after the HIV virus enters into the body, and then a general prodromal symptoms is going to take place following an asymptomatic phase and quiescent period, and then lymphadenopathy, hepatosplenomegaly is going to be involved. So non-specific symptoms related to enlargement of the reticuloendothelial system is going to take place, and usually it follows an incubation period, you can call it, of about 1 to 4 weeks, and then you are going to have a lymphadenopathy, as I said, and hepatosplenomegaly associated with that, and then we will see the here manifestations of the immune suppression scenario that is going to take place, and then the appearance of the opportunistic viral, bacterial, and parasitic infections that is going to take place. That will be followed by a sudden reduction or decrease in the number of T helper cells. And of course, here it talks about the treatment regimens that is going to take place, and the use of here the reverse transcriptase enzyme inhibitors that is going to reduce the non-nucleoside, we call it reverse transcriptase inhibitors, that we could use here for a longer period of time, and sometimes for life. That is going to reduce the number of cells that are infected, and we will keep monitoring the patient with the number of CD4 plus or T helper cells of those patients, and see how much they are responding to treatment. So here, this is the clinical scenario of the immune response and the development of viremia that is going to take a place. And usually, as you can see, the disease will progress usually after the fourth week to almost 1 year of a progression, where viremia usually will take care. It will take the presence of almost in 2 months time, and then it will go up, and then the antibody level, as you can see, will start progressing in those cases. So this graph will show the progression of the viremia state, and then the immune response related to the infection of the virus at the early stages of the disease, followed by the viremia, and then the immune suppression stage that will follow usually after here the second, third month here of infection, and then the immune suppression final case that will be manifested by the dramatic response of the suppression of the CD4 cells. So the final outcome then, and the indicator here of the progression of the disease has to do with the number of CD4 cells, as I said, and of course with the antibody response that will come out after the 8 weeks of infection. So then the progression of the AIDS disease and the pandemics that is usually caused by HIV-1 or HIV-2 that will lead to paralysis of our immune system. And then finally the development of the opportunistic infections. And then at the end, because of the immune suppression, then tumors could be here emerge, like the Kaposi's sarcoma, for example. So it's either then we see a rise of HIV antibodies, and have a reduction of the T helper cell lymphocytes, usually less than 200. So 200 usually is the marker level that indicates a severe immune suppression. And up to 500, where normally it's 800 to 1200 normally, but when it goes to less than 500, then here the patient has to be monitored. But when the number is less than 200, here then massive immune suppression is going to take place, and opportunistic infections will follow. So then the pathogenesis has to do with the infection of the T helper cells, and then the dramatic reduction of the number of the CD4 cells. And as I said, here when the level goes below 200, then a massive opportunistic infection will follow. And also, it's so important here to follow up on the chemokine receptors that are going to be involved, and wherever those CD4 cells or antigens are available in the brain, for example, the mesenchymal cells, oligodendroglial cells, for example, the enterocytes, and then you can follow those in general. Usually latency is going to take place inside of those macrophages, where it's the main reservoir of the infection. So the latency will take place in those, and mitogens or here antigenic activation is going to take place after here going to take place in order for infection to follow. So the macrophages then, they will act as the main reservoirs of the HIV infection. Sometimes we could have what we call cell-to-cell fusion and involvement, and the CNS is usually involved by the oligodendroglial cells of the CNS cells. So here the immune deficiency then state that will follow, that is related to the infection of the macrophages and then the T helper cells. So the killing of the T helper cells that are infected then, and then their removal here of the ones that are not infected by binding here into the ones that are infected, and taking out those non-infected cells as well as the infected cells. So the macrophages, they become defected, and then this is going to increase in the immune suppression, which leads to suppression of the cell-mediated immunity in general. Some of the cells, we call it polyclonal B cells, they will be activated, and antibodies could be produced during the process of infection. So T cells will go down, but B cells could be stimulated and antibodies could develop. So the clinical manifestations then it varies from silent to multiple opportunistic infections and cerebral damage. So we see three phases, phase 1, 2, and 3. In phase 1, for example, usually we see it in 2 to 4 weeks, usually after the infection, following the infection, and started with asymptomatic to mononucleosis like illness, enlargement of lymph nodes, lasts usually 2 to 6 weeks. And then we have phase 2, start with the long asymptomatic phase, maybe up to 10 years. And then phase 3, which we call it the full-blown picture of AIDS, the phase of the opportunistic infection. So it will be followed by the absolute number of T helper cells will go beyond 200 per cubic milliliter. So the opportunistic infections, which is viral, bacterial, or parasitic, it depends on the local area and what are the infectious agents that present in that particular area. So it depends on the locale, we said. So the common opportunistic infections and the malignancies associated with AIDS. First, we'll start with the protozoa, like Toxoplasma gondii, as well as the Isospora belly infection, and of course the Cryptosporidium parvum, for example. And then followed by fungal infections, like for example the Pneumocystis cerevisiae infection, and the Cryptococcus neoformans, and as well the candidiasis, Candida albicans, is very common in AIDS patients, and of course histoplasmosis dissemination, like Histoplasma capsulatum. And finally, in the fungi, the Coccidioides immitis that will be disseminated. And then we have bacteria like Mycobacterium, disseminated mycobacterial infections in general. Mycobacterium tuberculosis that will be disseminated. And also the Mycobacterium avium intracellular infections, and usually this is a non-virulent organism, but because of the immune suppression, it could cause dissemination. And then the viral infections in general, like, for example, the persistent mucocutaneous herpes infections. We see that. Cytomegalovirus, CMV virus, and the varicella zoster dissemination as well. Progressive multi-organ leukoencephalopathies virus, and the opportunistic malignancies in general, like Kaposi's sarcoma here, and lymphomas as well will be associated with the immune suppression state. So this graph was taken from the CDC, the Central Central Disease, the CDC, the Center for Disease Control in Atlanta, Georgia, showing the number of cases. It has been discovered in the late 90s, and the dramatic increase in the diagnostic abilities to diagnose, and then the decreased number of cells over the years because of the improvement of and the discovery of the antiretroviral medications and treatments. So improved improved prevention, and improve treatment regimens, that they become very, very effective. So you can see in the late 90s, the increased prevalence of the cases of the HIV, and then how the improvement is getting here much better related to the diagnosis and the treatment of those cases. So this slide also shows a comparison of the most common chronic illnesses in the United States compared with the HIV infection. And you can see in the graph, for example, in the early 90s, a very large number of cases of AIDS. They were on top of the list of the number of deaths or mortality rate of all chronic illnesses, and how they managed to drop out here because of the high progression diagnosis, treatment, and prevention of the HIV. And you can see and compare of these chronic illnesses with the HIV to see how much improvement here we got. Here improvement of our health measures related to diagnosis and prevention and treatment of the HIV. This is in the United States, but this is also all over the world. So in making a diagnosis of the HIV virus is usually done by serology, and the enzyme-linked immune sorbent assay is very specific, but it could be associated with false positive results. They have discovered what we call the Western Immunoblot technique, and in the Western Immunoblot technique, it's usually in a confirmatory test if the ELISA is positive. And usually the first 2 to 4 weeks, here we could get a false negative results, and this is what we call the window period. And so here we have to confirm by the Western Immunoblot technique. And the antigen that we are looking for is the P24. So the P24 here and the anti-P24 antibodies are the most important here antigens to follow up. So the P24 is usually appears first, and then the antibody will follow afterwards. So the detection of the P24 is extremely important if it has been managed or discovered early of the infection. So the confirmatory test that we do when the test is ELISA is positive is what we call the Western Immunoblot technique. And the Western Immunoblot technique, just simply we get the virus through electrophoresis, and then we do an immunoblotting. That means here the viral antigen will be transferred into a nitrocellulose filter paper, and thus the nitrocellulose filter paper they will be cut and stored. And so the nitrocellulose filter paper they contain antigens that will represent the different structural components of the virus from the surface antigens, for example, into the structural component, the GAG, the ENV, and the POL, for example, antigens. And then we get the patient serum that's positive with the ELISA, and then we incubate it here with these paper strips of the virus antigens, and then we add a secondary antibody with an enzyme added into it, and then you will see here the appearance of bands representing the different antigens of the virus. So the interpretation of the Western Immunoblot technique, that here we need to have a band from each component. As I said, here the structural components, the enzymes and the surface proteins, and we have a positive control and a negative control. And from there you can confirm the diagnosis of the HIV. So the treatment, many antiretroviral medications has been discovered. The problem with these medications that they have to be used for a longer period of life, maybe lifelong treatment. Maybe the oldest drugs were the AZT, the azithromycin, for example, and those usually is not effective against the integrated virus. Then they have discovered the nucleoside, the reverse transcriptase inhibitors, and those were very efficient when they have discovered, like the dideoxyinositine and the dideoxycytidine. And then then the non-nucleoside reverse transcriptase inhibitors, and they believe that those are very, very effective in the treatment. So then they discovered the protease enzyme inhibitors, the glycoprotein 41, for example, inhibitors, like the GP41 fusion, they call it inhibitor, like the enfuviritide, for example, and they say that this is very effective. And among those also, we have the ICR5 inhibitors. They call that the Marvi rock, and the integrase enzyme inhibitors. They call that the raltegravir. They say that this is also very effective. So most of the enzymes that the virus uses for replication, they will be inhibited by these medications. And this drug, usually we use it in combination in what we call the highly active antiretroviral therapy, and they believe that this combination of therapy is very, very effective. And then the final stage is to treat the opportunistic infections, bacterial, antibiotics, antiparasitic medications, viral, antiviral medications, and so on. So this table shows the most common antiviral medications that are used for the treatment of HIV infections. Those are the currently FDA approved, and the Department of Health here regimens that are used here in combinations, like the nucleoside, for example, reverse transcriptase inhibitors, as well as the protease here, transcriptase inhibitors, and of course here the protease inhibitors, and the integrase inhibitors, and the CCR5 here antagonists, for example, and the GP41, which is the fusion protein inhibitor, and a mixed combination of those. So those are the most common medications that are used for the treatment of AIDS, and they believe that those are very, very effective. But the problem, as I said, that these combinations of antiretroviral here medications, they have to be used for a longer period of time, which could be for life. And they say that they are extremely effective, and those we use when the number of, as I said, the T helper cells drop below 200 per lambda in general. And when we use these combinations, here the patient could really improve and almost lives a normal life. So the prevention, then we have to look into how people they do get infected, usually through what we call a promiscuous sex. We mean by that people that have more than one sex partner. We mean by that prostitution. So control of sexual promiscuity, and sexual promiscuity is prohibited all over the world. So we have to educate people about that, and in order to control this, and this is going to decrease the number of cases if that is really controlled. And the second mean is by drug abuse, especially here the ones that they use here one syringe with multiple partners. So the control of the drug abuse, also we can control AIDS. And that the people that are working in laboratories, they have to stress here the significance of laboratory safety, especially here by accidental needle injury, and how to or how to get rid of needles in the lab in general. And what about immune prophylaxis and the use of vaccines? Nowadays, people are still working on an effective vaccine against the HIV virus. Till now, we don't have any dramatic here discovery related to that, but most research they say that they are promising and waiting for a general vaccine for this. Second thing is about chemoprophylaxis, especially in those that they are working in the blood banks and so on. They could take antiretroviral medications for prophylaxis, so they won't be infected.